All right, welcome everyone. I'm very happy and excited that you all made it here to this very unique and special meeting. I especially like to welcome you, Lynn Hershman Leeson, for being here. To give you a brief background on how this whole came together, a couple of months ago, Sabine Himmelreich from the Haus der elektronischen Künste here in Basel, she's the director, she contacted me. And she is interested in our projects, in the Art of Molecule projects, and she said that they're going to have an exhibition coming up with Lynn next June here in Basel. And Lynn is always working on her exhibition, especially on this one. It's an exhibition that always develops and evolves. We already talked about this a little bit. And so she said it would make sense, would be interesting that if we hooked up. So Lynn and I had a couple of emails and Skype meetings, and I'm glad we made it here together. Yesterday she was visiting Novartis for a whole day, and she told me that actually they're going to name an antibody after her.
No, really?
Yes, so that puts us under pressure.
It was their idea.
This puts us under pressure today, so we need to top this somehow.
It's going to take 3 months.
That's it. Well.
So before I start talking about our work, I want to embed it a little bit into philosophy, because I think when science and arts meet, then somehow philosophy is in there as well. And it is also what is kind of motivating me to do the research, and I think lots of us to do this kind of research that we do. So, but for this, the question is, what is actually research?
Just wanted to start off with a thought here. So this is a drawing of a 1,000-year-old drawing of a brain, and specifically,
Just explain.
yes, specifically, it is actually a diagram of the visual system itself. So I don't know if you can
And
see
you have
this.
the nose in the center, right?
Exactly, yeah. So some things are vaguely familiar. For example, you can see the eyes, and you can see the optic nerve where it goes to, sort of back. And as Daniel said, a nose that essentially connects to, well, nothing.
It's the eyes. I don't know.
I think you guys will all agree that our understanding of the visual system has gotten substantially more complicated.
So yeah, that's the instrument I've been talking about. So it's here hidden in that box. It's something that is really customized. So far, nothing like this on Earth exists, and it's it doesn't look like much, I would say, right? I mean, it plus some cables and some things stick together, but that's how it works. But there's lots of electronics behind that needs to control all those things there. So I mean, we have several computers, like 2 computers here, and you see here like the whole tower here, like this instrument, these instruments, all that. What you see here basically belongs to that system in order to control everything that is going on inside.
And how much does the cell weigh, did you say?
So roughly 3 nanogram or so. It's like a 3 billionth of a gram. So really tiny. It's somewhat hard to imagine, right? I mean, I still have no imagination for that, right?
Yeah.
I mean, it's just too tiny somehow. But that's the good thing. I mean, somehow like that is what I meant, like that with this technology you basically extend your senses, right? You cannot measure it by just touching it or lifting it up or something. It's just impossible.
Yeah.
But now we have those tiny cantilevers, and they can touch them and lift them and feel the mass, basically, right?
Wow.
That's how it works. Yeah. So right now there's also the setup of Rajiv, so maybe he wants to talk a bit about it.
Sure, yeah. So this is the project that I was talking about. I only gave it a single cell application, so what we can do is here you see on the video is we mount a pipette there, and we have our dishes of cells, so there's some cancer cells there, for instance, and then we have our magnet where we can essentially take our viruses and pretty much guide it to any cell that we want of interest. But in my presentation, what I'm showing you here is only a single cell application, but we've gone a step further now. Unfortunately, I can't show you the mouse facility, but one thing we can do right now is also electromagnetically guide viruses to anywhere in your body. So that's another project we're doing pretty much. So for example, we have a mouse right now they've generated, it has a melanoma tumor, and when we do a systemic injection, and then we use magnets, it just takes it straight to the tumor there. And
Wow.
yeah.
Have you
And
seen differences with the stamping?
Yeah, for sure. So we've combined the power of viruses, where viruses have evolved over centuries and eons to essentially go to specific cell types. For example, HIV attacks your immune cells. So the lab that I'm working in, they have generated viruses that go specifically towards cancer. Melanoma cells they've generated. They've just engineered some of the variants of this HIV virus to now attack melanoma cells. But the problem was for a long time was that if you inject it, they still, they just diffuse all over. They're not guided to their ultimate target. And so we saw significant differences when we could essentially take those viruses now and guide them to their location. And and we're actually also doing some imaging with that. We actually are doing whole animal imaging. So I don't know if you've heard of this technique that came out of Stanford a while back. It was Clarity. It's Carl Dijsselroth. I can show you later maybe on my computer. But so we have taken it from cell culture to mouses' brains, where we can guide these viruses and specifically direct any gene editing, fluorescent proteins, as you saw, optogenetic proteins, where we can control these cells via light now. And also now Danny's looking at another project to extend this now to control them electromagnetically. So meaning you can electromagnetically turn on your cells on and off.
Wow.
Yeah, so these are, and I can show you if you want to see the actual imaging that we do. There's also the other, so I can lead you outside and quickly.
Just one thing that just came to my mind as well, because we were somehow talking about sound, and it is like that actually we have an additional source of sound, because every time that we move something, you might want to hear, you want to go there somewhere, and then you can listen. I actually was wondering whether I should write at some point a program, because when we move, those things, and it's not live right now, but then a sound appears. You hear that one? And when we move a little slower,
Goes down.
that is basically slower.
Well, that's the machine. And
That's the machine.
The cell must have 2 to 20 hertz, or makes this it makes this more this. Was heißt der Ton of the monks? It must have this frequency because it's 2 to 20 hertz. We have to transfer that into a real big bass. I want to hear our 2 cells singing.
Probably
We'll make a chorus.
The
last
no
waltz.
problem to do.
We don't sing our life. We let the cells doing the work. It's much easier. He's a Nobel Prize chair committee, and some people are just, and some just whoever. It's I only have that one. Whatever you want to sign, however.
I always have to tell the master students it's not for everyone to sign, because there are some guys drawing some stick figures on it or something.
Thank you.
Sure.
Great. Thank you. We're happy. So you showed in some pictures.
Yeah, some images, and then it's become a big source now. It's for so this is so here it is. So what we do is we image the whole brain itself in its intact state. Yeah, so we can actually go in, and so I can show you some of the videos that I made for this. And so with viral stamping, you can actually see in its full 3D context a single neuron.
Wow.
And you can see how it's behaving amongst hundreds of neurons firing, and so this is one of the things that we're incorporating. And also for tumor imaging, we want to see, for example, where the cancer cells are, how are they moving in 3D space in your body itself.
Wow.
So that's one of the
Amazing.
There's a common problem in biology, and oftentimes you want to understand what's going on in a cell, and in particular with what genes are being expressed throughout the life of a cell or in different developmental trajectories. And so what people will do is they'll set up one experiment, but they'll set up 8 different groups and then collect it at different times. And so this tells us what happens in a batch, but not necessarily what happens in the life of a single cell. And so Florian's project is about developing the tools to allow us to do this on a single cell level.